Hi everyone, here's Chocolate, here's Fluffy. And I got a letter this week from one of you in the wonderful audience, and they said, Linda, in these strange separated days of COVID, isn't it interesting how the animals that we have lived with, maybe for years, and we have loved, and all of a sudden, when we must stay away from people, our animals are beginning to be like our friends, our family at the deepest level, at an even deeper level. And it is, as I thought about it and read the letter, I have loved these little guys and I have loved other animals that I've had throughout my life. But something is changing in a world where we know that we're never going to be in the same planet that we were, say, at Christmas time. That everything has really changed. And that, as I read that letter, I thought, I really, truly, in my heart and my soul, I love these little guys who are like filling something that you never ever associated in your life, that you would have such uncertainty about what was coming every 24 hours, that you would feel a strange disconnect even about what is life going to be like now. And these two wonderful guys, Fluffy and Chocolate, they make me laugh, they make me run to get a ball, and when they make me do that, some part of me returns a little bit to that world we had at Christmas, and I don't think we'll ever have the same again. And so I am seeing that my dear Chocolate and Fluffy, as the letter writer, and I'm sure a lot of you, they are my connection to the world pre-COVID, during COVID, and will always be after COVID. And I have some really interesting things to share with you tonight. So I'm going to let them down. I love you guys. And in these very serious days of COVID, I got a video this week by email that made me really laugh. The video is genuinely funny. It's from my daughter Laura in Philadelphia, who was in the kitchen with her iPhone when Hannah, my 5-year-old granddaughter, climbed up on a chair near the kitchen sink. What happened next, I would like to share with you. See if you can figure out what Hannah is reacting to.
That's not gross, Shadow. What
What?
I'm about to do is going to be gross. That wasn't gross, bro. What I'm about to do is going to be pretty.
Let's see what you
Ew, that's blood. I can't watch. Oh. I can't watch it.
Okay, you'll have to.
Told you.
Yeah. Oh.
You're so good.
Wait. Ew.
Don't fall over.
I told you it was going to be gross.
I can't watch. This is too
You want to come with mommy?
I'm just kidding.
Whatever you think Hannah was reacting to, let me know in the live chat now. Peggy is going to keep track, and in the last 5 minutes of tonight's live stream, about 55 minutes from now, we'll give you the answer. And whoever is the first to guess correctly what Hannah was reacting to, I'll send you an autographed copy of my book, Mysterious Lights and Crop Circles. and to give you time to interact with the live stream chat now. I'm going to give a cheer to the Hubble telescope. NASA launched Hubble 30 years ago on April 23rd, 1990. So Hubble's 30th birthday is tomorrow. And I think one of Hubble's most astounding images was first photographed on April 1st, 1995. It's the Eagle Nebula, and this is the original photo that you've seen everywhere around the world, even on t-shirts. It's also known as the M16 Nebula. It is about 6,500 light years from Earth, and this April 1st, 1995 image was taken at Arizona State University. But the public did not see it until NASA's November 2nd, 1995 press release, which said, quote, "These eerie, dark, pillar-like structures are actually columns of cool interstellar hydrogen gas and dust that are also incubators for new stars," close quote. And more recently, Hubble took this new image of the Eagle Nebula with a combination of near-infrared and visible light. That enhances the nebula's sort of otherworldly beauty, where newborn stars are glowing inside of those amazing cold gas pillars. Another amazingly detailed Hubble image was made in 1999 and 2000. This is the Crab Nebula, also 6,500 light years from Earth, which is a supernova remnant in the constellation Taurus. At the center of the nebula lies the Crab Pulsar, a very dense neutron star only about 19 miles across, and it spins at the rate of 30.2 times per second, emitting pulses of gamma rays and radio waves. Now, in 1995, Hubble pointed its extraordinary mirror camera at an empty patch of sky for a 10 full-day exposure. At the end of those 10 days, here is the resulting image of thousands of galaxies going back eons in time. Even the smallest points of light in this image is an entire galaxy. The tiniest points are entire galaxies. From Hubble's what they call deep field study of our universe, astronomers now calculate that there are 2 to 3 trillion galaxies in this universe containing 1 septillion stars. That's a 1 followed by 24 zeros. And another way to say it, there is a trillion stars. This beautiful Hubble features the bright red supergiant called V838 Monocerotis. It's 20,000 light years from Earth. Back in 2002, mysteriously, the red supergiant became the brightest star in our galaxy, and no scientist knew what had happened. But Hubble captured this gigantic brown cloud of exploded gas surrounding the beautiful red giant star on February 8th, 2004. Okay, I hope you've had fun guessing what made my 5-year-old granddaughter, Hannah, cringe in the kitchen. And near the end of this live stream, I'll show you the answer. And then Peggy can tell us if anyone guessed what was happening. And I want you all to know that it was nice. It sort of lifted my heart against everything I'm studying in COVID to open tonight with Fluffy and Chocolate and Hannah and the glorious beauty of the Hubble images. Because sadly, it looks like the world is going to have the COVID-19 coronavirus pandemic through this summer and into the fall. And that's when there could be a second wave of this wicked disease that medical experts say could be much worse the second time around. In fact, that's what the director of the Centers for Disease Control and Prevention, announced today, quote, "There's a possibility that the assault of the virus on our nation in winter 2020 to 2021 will actually be even more difficult than the one we just went through. We're going to have the flu epidemic and the coronavirus epidemic at the same time," close quote. And that's what happened in the 1918 flu pandemic. That first pandemic wave was mild compared to the second wave, and that second wave killed 50 million people. Spanish flu was a DNA influenza, not an RNA coronavirus like COVID-19. So what are RNA and DNA? All life forms are made up of genetic codes. In humans, we have DNA codes and RNA codes. Our biology functions on DNA controlling the synthesis of proteins and using RNA, which is ribonucleic acid, present in all living cells, to act as a messenger to carry out the DNA instructions. So essentially, RNA and DNA, under normal circumstances, are collaborators. If you are a genetic scientist working in a lab trying to understand the COVID-19 coronavirus and to create tests to find out how many people actively have the disease or don't, in order to separate out the infected from those who do not have COVID-19, you are working in this world of gene sequencing. The yearly flu bug is a DNA microbe. The new highly infectious and deadly COVID-19 is an RNA microbe. In order for scientists to come up with accurate tests for the RNA invader, they use RTPCR. That's the title at the top of this graphic. It stands for Reverse Transcription Polymerase Chain Reaction, or PCR. And all of this is why you aren't hearing details on television or in podcasts about why there aren't enough COVID-19 tests in the United States, which has a current population of 331 million people and needs at least 300 million tests. And one of the big reasons for why we're never hearing a reason for why is it so hard to get the tests. It's extremely complicated. To help me understand more about the insidious COVID-19, I contacted Professor Jay Couey, Ph.D. He's a cellular neurobiologist at the University of Pittsburg in Pennsylvania. He earned his Ph.D. in 2009 and went on to do a postdoctoral degree at the Norwegian Institute of Science and Technology, where he was a research scientist there for 5 years before returning to the United States and teaching at the University of Pittsburgh. First, Dr. Couey explains the challenge of testing for COVID-19.
Reverse transcriptase PCR is a reaction that everyone who works with this reaction and uses it for scientific measurement understands that you need to do a whole bunch of controls in order to rule out false positives. So let me just give you an example from my own work. Mm-hmm. I tried to pull individual brain cells out of a brain slice and then do reverse transcriptase PCR on one neuron to see if it expressed the mRNA for nicotinic receptors for acetylcholine. So just to boil that down to easy words, I took neurons out of the brain and I tried to use PCR, the same test that they're using for coronavirus, but I used to test for a specific mRNA for a specific protein. In order to do that and publish it as a positive result, I need to do 3 different pairs of primers. Those are the little sequences that are supposed to be specific for my molecule that I'm trying to detect. I need to have 3 different pairs of those so that I have to get positives on all 3 of those pairs. Otherwise, people will automatically assume that the one that is positive just happens to react with something else. So here's already 6 tests, right? Because if I have 3 sets of primers and I have to test them on neurons that I think are positive and neurons that I think are negative, then in the ideal result, I will get 3 positive tests from the neurons that I think are positive and 3 negative tests from the neurons that I think are in the group that are negative.
What did Germany and South Korea use?
Well, they're only using a test with 1 set of primers. So it's already way below the threshold what any normal molecular biologist would consider the bare minimum to say that you have a positive test. And these sequences are only, let's say, 100 amino acids long. They can't be too long because then they'll just wrap over on themselves and they won't amplify. You're kind of limited. You could never use a primer, for example, that was the whole sequence of the virus. It has to be a very short piece and attached to something else. This shortness, by definition, limits the specificity.
We have seen all kinds of people being interviewed on TV news programs saying, I was tested positive for COVID-19. What does that mean, and what was the test?
My guess is that that means that they swabbed their nose and they swabbed their throat and they ran a PCR test with 1 or 2 sets of primers, and then they got a positive result. And remember that in any other scientific paper, at the very bare minimum, you would need to do 3 of those tests with different primer sets. At best, what they are doing now is a single test, and if they get a positive, it's a positive, and they put it in the bucket. And I, as a molecular biologist, would have had to get 3 positive tests from 3 different primer sets, and then I would put it in the positive bucket. Anything else would be a negative. They're not doing anything like that now. Every positive is a positive, and they don't care about false positives, which is absurd with PCR. Every immunologist agrees that the antibody test is much closer to flawless.
Okay.
But then you have already had it, and that's the problem. There is no way for anyone to be sure that you don't have it. You can't prove a negative. And the positive, well, if you want to think from a safety perspective, you just have to quarantine everybody who's positive. But the point is that if you're not testing everyone, but you're only testing the people who are very symptomatic, then the statistical understanding of the pandemic cannot ever manifest.
What about the discussion that there could be a second wave, third wave? And just like 1918, it was the second wave that took out 50 million people a century ago.
And the reason why we're going to get second and third waves is because there's too many of us who haven't had it yet. So every time you give a virus a new population of uninfected people, you're going to get a new bloom. And the epidemiologists who have followed these things year on and year out have realized over years and years of observing it that the way that these outbreaks of respiratory virus are stopped is when the virus finally gets to the school, because the kids and the parents all get immunity, and then the virus can no longer spread. And that's the point. If you are immune to COVID and you get it again, you can never become contagious because the viral load never gets high enough. That's what immunity is. And this is the idea that you can test that you're accurately keeping track of who's sick, and that's the problem with asymptomatic spread. We keep the people who are contagious contained, and everybody else, you're building up immunity in the meantime. And this can't happen unless we know who's sick. And because we've been so lax in terms of finding out who's sick for whatever reason, we have not been able to get it under control like the other countries have. They're making it seem like you have blueberries in your throat and we have to look to see if the blueberries are there. You have the blueberries. No, it's not like that, because we have lots of viruses to test for. There's lots of bacteria there. There's lots of crap that can give false positives. And so we constantly need to refine the test, and we need to double check that the test is testing for what we say it is.
Right. And I'm hearing an irony that Italy has gone through hell, but they may end up with more herd immunity, and that we go into a second wave in the United States and other places, and we may have maybe even a worse second round, but Italy might actually be better off because they went through hell the first time around?
Yeah, it's very possible. But remember, none of us have ever seen something go through the population of this size. For the first time in maybe human history, a virus has enough humans that it can get all the way around the planet. And if there's enough humans in between you and the next time that the virus comes around, it will be, by definition, different enough to attack you again.
So, what causes a second wave of infection that is worse that second time around? And why are COVID-19 tests and antibody tests so hard to get when there is urgent need to separate people testing positive from those testing negative? And why can't we get a COVID-19 vaccine sooner? Scientists have tried to develop a SARS vaccine since the epidemic erupted in 2003, that's 17 years ago, but there still is no SARS vaccine and no MERS or MERS vaccine, which is another coronavirus that erupted in the Middle East in September of 2012. That was pretty deadly, but it was not as contagious at all as COVID-19. How does COVID-19 hang out inside some humans for 2 to 4 weeks? One case, they thought it was 6 weeks, hijacking the human host cells to copy huge amounts of the invading COVID-19 without the host even having symptoms of disease. Part of the answer. All of those spikes are made of protein that sticks out of a sphere that is packed with genetic code. The RNA code programs COVID-19, and each protein spike has a little sugar molecule at the tip, which wiggles and makes the molecular outline of the shape of this coronavirus difficult for the human body's immune system to precisely copy that ball with lots of spikes pattern in order to search and destroy the COVID invader. Worse, COVID-19 spikes are so close together, they snag human antibodies that are trying to fight against the invading virus. So many human antibodies get stuck in the spikes that they cover over the true COVID-19 shape. Adding to the problem are those sugar molecules that are shaking at the ends of each protein spike. It's very hard for the human immune system to figure out exactly what the invader shape is and to fight it. And the COVID-19 spiky pattern confusion also makes it more difficult to produce a vaccine. And all of this is happening at 1/20,000th of a millimeter scale, where only electron microscopes can actually, quote, see, close quote, COVID-19. Dr. Couey thinks the antibodies stuck in the COVID spikes could be one of the reasons that COVID-19 can hide out in people for so long without causing immediate symptoms to the host, while it's spreading to lots and lots of other humans around the host. The human antibodies can actually get in between the spike protons.
The antibodies can actually get in between the spike proteins of a virus, and rather than attach and then form a flag that says, hey, immune system, come over here, the antibodies fill in the space between the spike proteins and get stuck there. And then the virus, instead of presenting its outer coat that the immune system should try to recognize, it's presenting an outer coat that's almost completely composed of antibodies, antibody clumps. And that means that when it bumps into immune cells, if the antibodies are in the wrong orientation, the immune cell will take it in, but be unable to identify that this virus is foreign. And the reason why is because it now is going to make antibodies for the virus covered in antibodies instead of making antibodies for the virus.
Wow.
When the virus is covered in antibodies, the immune system now makes antibodies for a shape that is more akin to the antibodies that are stuck to the virus than the shape of the viral protein coat. What results is that those antibodies go around and they only attack other antibodies. So antibody-dependent enhancement has been known to happen for a very long time. First discovered during the attempted vaccination of people in Africa for dengue fever. Dengue fever is essentially a virus that changes quite rapidly, and so what happened was the children who were breastfed by women who had previously had dengue fever, so they had antibodies in their breast milk. But then these kids, the next year, would get a different dengue fever, and the antibodies from their mom would help the dengue fever to be even more devastating, and they would get what's called a dengue fever shock. So it was because the antibodies weren't specific enough that they ended up masking the virus instead of labeling it.
Is this why this COVID-19 coronavirus, there's been so much confusion about why isn't it not clear that a person has antibodies? It hides.
Yeah, and I think that this comes down to a specificity of these tests. But this coronavirus is very special because, number one, you get a very high viral load before you show symptoms, if you show symptoms at all,
Right.
which is not typical for the common cold. It's not typical for SARS. It's not typical for MERS. It's not typical for any.
Right.
It's a special one. It's a special one in a devious sort of way. One of the arguments that this came from a bat is because when they have gone to villages in these regions of China where these bats exist, there are people who have antibodies to these viruses already, which would lead any immunologist to say that these people have had this virus already, and that's why they have antibodies to it. It's just that if this gets to a big city like Wuhan, then it will explode because nobody's got immunity to it. Now, if it's a laboratory virus, remember, that would mean we know they did this. They went around and they collected as many of these viruses as they could. How in the world would they be able to come up with all of the different viral sequences that they've compared this to if they didn't go out, collect those viruses? And you can't just take the blood of a bat and then the viruses fall out. You have to isolate individual variants of the virus, and you have to grow them in sufficient quantities in cell culture so that you have sufficient quantities of identical viruses that you can then sequence. You can't just pull out a single viral particle and sequence that one, and then pull out another viral particle and sequence that one, and then now put those on blast, and then that's it. You need to make quantities of each of those viruses so that you can sequence them. That means that there somewhere, at some point, was a vial full of virus X, virus Y, virus Z, all the way up to every virus they've ever shown you on a sequence list. Every pangolin virus, every bat virus, every mouse virus, every rat virus, every canine virus, every virus that has ever been sequenced and shown in one of these papers that's been recently released, that virus has been sampled in the wild, amplified in a laboratory, grown in cultures, selected in culture, and then sequenced. So already then, if that virus turned out to be a pandemic potential virus, it's already in a much more dangerous situation than it was when you took it out of the animal in the wild. Because it's amplified from being one or two copies in that animal to being millions of copies in a laboratory. You cannot say that this couldn't have come from a laboratory.
Right.
Because we've had laboratory accidents before, and because all of these papers exist, these viruses exist in a laboratory in quantity. And because they exist in quantity, alive and virulent, if one of these has the potential to be pandemic, and you have one idiot or one negligent or one careless or one very evil person. That's all it would take. And viruses are nothing more than quite complicated protein, which has some genomic material inside of it.
So you wouldn't be surprised if, in the end, that it is established that this coronavirus, COVID-19, could have been, let's say, in that Wuhan biohazard 4 lab, not necessarily for any bioweaponry, but was part of sequencing all of the coronaviruses since they had to deal with SARS back in the beginning of the 21st century. And it would be logical that in labs like the biohazard 4 in Wuhan, that they would have done a lot of sequencing of coronaviruses, and something could have happened in the lab that got loose.
Yes, it's plausible.
Given the intricacies of appropriate and professional laboratory working with coronaviruses, trying to come up with ways to defeat those that are dangerous, like SARS and MERS and now COVID-19, and in the process could create this insidious monster that we're trying to deal with now, why is it so hard and so long to get a vaccine when we know so much about the coronavirus?
Well, that's the worst part, right? We've been trying to make a vaccine for SARS since it came out in 2003. The Chinese have been working on it since then.
But why is it so hard?
Because the antibody, that's the problem. So the antibody-dependent enhancement effect is most often seen during attenuated vaccination. When you attenuate the virus, when you partially digest it with formalin or something else, and then give that attenuated viral particles to a person as a vaccine, those are the people the most likely to produce the antibodies which lead to antibody-dependent enhancement, ADE. The ones that survive the live infection almost never display ADE. So this is, again, where the vaccine, in my mind, they already know they can't make one, because every time they've tried to make it, they've had this problem with antibody-dependent enhancement, because the weakened virus doesn't end up evoking an antibody response, which leads to immunity. The other problem is, and this is another thing that they aren't adequately addressing, is that if you have an effective antiviral drug like remdesivir, and lots of people are now claiming is effective against SARS and effective against SARS-CoV-2, that if you use a drug to prevent virus from replicating, you also prevent the actual immune response. And so then your body never builds immunity.
Oh, wow.
So one of the reasons why a lot of people becoming infected again in Wuhan and again in South Korea, this is my armchair guess, is those are the people who received antiviral drugs very early in their presentation, and so they never actually developed an immunity, so they're still fully vulnerable. And that was already shown with MERS. There's at least 3 papers that showed that there were recurrences of MERS in people who were treated with antivirals because their antibody titers were at 0 after a year. I think it's more likely that we're going to go and get antiviral drugs to work simply because the vaccine has proven so biologically difficult to pin down.
Wow.
You know, it works with certain DNA viruses and it works with other viruses. Again, because the immune system is so mysterious, we're just not really sure why it's been so hard to do a vaccine with coronaviruses, but it's probably because of their shape.
The shape.
Their shape. These spike proteins are very difficult for our immune system to deal with. And so we are more likely to develop a symbiotic relationship with them like we have with the common cold than we are to eliminate them.
All of that grew out of my frustration that when I tune in to TV news, I have been feeling like I'm getting more politics than I am honest insights into science and medicine often. And I wanted to talk with somebody who didn't have any political axe to grind, who works at that level where you would say that COVID lives. And that when I did the first call with Professor Couey, my idea was that I would just talk with him so it would be educational and make notes. And I asked him if I could record it so that my transcript would be accurate, and he had no objection. And that discussion last weekend went on for 2 hours and 6 minutes. And when it was over, I realized that I was finally beginning to get a little better understanding of why it's so hard to do a vaccine, why the testing, the complexity of it, hearing him talk about it from an experiential base. No one telling us in straight language, this is tough, these are the problems, this is why, and finally beginning to understand. And that perhaps the worst of all is his confirming that he expects not only a second wave of COVID-19 this fall, but a third and a fourth. And depending upon what happens with the virus as it goes into human bodies and comes out and goes into waves, we just don't know yet, because there aren't enough people who have been able to be tested with antibodies, which he said it would be antibody testing that would actually be clearer and more accurate. But even that is a problem. And so I hope you don't mind that I brought from my 2 hour and 6 minute conversation with Professor Couey 2 segments for you that are normally, they're longer than I normally would have done, but it seems like we are at such a crucial moment in the timeline, and I feel always that the more we can understand, the better off we are. So I hope that you will take all of this in with the idea that nothing is ever going to be the same again, and that if a second COVID, another wave, is coming this fall, winter, we're going to be in this state of separation, no matter how many business people want to be pushing open doors. We all would love that, but not at the expense of our fellow human beings and people that we love in families and our dear friends. No, this is a tough one. And until they come up, maybe remdesivir will help, but as he said, it's an antiviral. And they have found that people who have taken the antivirals in the beginning, they don't build up antibodies. So we're in some kind of a new world where everything is upside down and backwards, from the macro down to 1/20,000th of a millimeter. And I'm going to try as hard as I can to keep trying to bring to you what I hope is important information for you to understand. And now, Peggy, I would like to take a few questions, and then at 8:25, I've asked Brad to give me a time cue, and we will then answer, show, reveal what my 5-year-old granddaughter was reacting to. So, do we have some questions now?
Thank you, Linda. First, I would like to say some thank yous to our super chats tonight. So thank you, Moonbird, Barry Marlowe, Mike Martin, Stephen Ely, Michael King, Jeffrey, Cat Chaser, Buzz, Terry M. Hart, Minnesota Folk, and Derek Bossler. Thank you so much.
How do all of you come up with your wonderful, rich names? I love it. Thank you so much.
We have a great question, and someone was wondering if you've noticed an influx of people reporting UFO sightings over the last 30 to 60 days because more people are restricted to their homes.
If you had asked me that question a month ago, I would have said that a video that I had gotten from England that was not nearly, we didn't know how we would show it here, was very faint, but it was definitely pairs of lights. And then I got another pairs of lights. I think it was from Washington. I got one from the southeast. I was getting videos from iPhones of objects that seem to be in pairs or in patterns. And today, as we are at April 22nd, Brad has an app for Elon Musk's, the little satellites that go in lines. And what we're finding, he did where the times and the dates where I was getting these reports, there seems to be sort of a matchup. And none of us realize that the Elon Musk launch, Brad, it's now in the 5th or the 6th today was a launch. The 6th, I think it was. I think it was the 6th launch today. Now, we're not hearing about it on news or anything because it's like old hat, but Elon Musk continues to put up these little satellites, and when they go up, they start out in a straight line and then they get further and further apart. But they do end up, I've seen a NASA thing, they literally swoop with pairs. Yeah, this is the release today. This is what released a whole bunch more of those little satellites today. And they will come out, and they will come out into long lines, and they are in the beginning equally separated by distance. And then as they continue to go around the Earth, the illustration that I saw in NASA this week, you will, if they started out like this in a line, they end up making a kind of spirally pattern. And they do kind of have pieces that look like maybe there's pairs and other combinations. So now we have another challenge. How do we tell if something for sure is Elon Musk's little satellites or something that is another intelligence that might now mimic Elon Musk's little satellites and be totally camouflaged in the night sky because we would all say, well, that's Elon Musk's little satellites? Or those two, and then a third. Would the lockdown in the various points around the world, if there had been a lot of UFO activity, would we have been able to notice it? Well, there have been some, what I would say would be craft reports, not little satellite reports, but not a huge flurry. And then there is this other interesting question. Since it's very true that the intelligence is interacting with this planet.
I'm going to interrupt. I'm getting out. I'm going to restart.
Okay.
And it'll just take a minute. Everyone, hold on.
Isn't it interesting that we would have that kind of an interruption for reasons unknown, right when I'm trying to talk about what might or might not be in the UFO category versus satellite versus maybe something else. And what I was just going to say, since we know that we have had competing extraterrestrial civilizations competing with each other on this planet and doing so with holograms and neutralizing gravity and especially invisibility in and around human populations, wouldn't it be natural that in this time where we're putting little patterns of satellites around the world and going into more and more and more out there, that if you were one of these non-human intelligences that monitor our planet, you would know that we were being inundated by the coronavirus and that people were staying home, and therefore people who would go out with night vision or infrared on the sky might have a chance of nailing something that is always camouflaged by the sky at night, meaning that's the camouflage. So there's no clear direction for me in terms of, are we dealing with an increase in activity outside of the confusion around the little satellites and the possibility that mimicking after mimicking after mimicking, that we have to always be thinking, well, what could be camouflaging itself as? A drone, a plane, a satellite, and is not. But I don't know if we humans, outside of intel and counter-intel and all of the sophisticated technology they have, I don't know that there's much of a way for us humble humans to know for sure without collaborating with somebody who really has sophisticated technology. And I love working with people who have really sophisticated technology. So if anybody out there does, or they want to try to take advantage of April to May when at least some of the world is still going to be in lockdown, it would be interesting to see what is in the sky now. Okay, what about another question?
A viewer asks, what are your thoughts about the moon being hollow and perhaps the place for lizard beings?
No question in my mind, since I have had half a dozen people who have worked in military, intel, the Pentagon, who have told me all the way back to the 1980s that our government has known for a fact that this moon that orbits our Earth is responsible for the tides. It is hollow. The words that have been said to me, it is an artificial machine. The moon is an artificial machine created by one of these advanced intelligences, placed to monitor and come and go to Earth without basing on Earth, and that there's all kinds of advanced technology inside the moon that has been protected by something. I don't know if humans have ever yet penetrated to what has been described to me and sketched for me, and even a photograph was shown. And the problem is, how do you prove it? Because people who come from intel and counter-intel with what they say are their first-hand experiences, Antarctica, the moon, Mars, and interstellar trade, they either don't have access to, or they just don't want to risk. Once you start moving into evidence to an investigative reporter, which would be videos and photographs and maps and illustrations and computer printouts and all of that, it's a whole other ballgame. And that then somebody like me would then want to take an accumulation of all of this physical evidence and lay out chapters about what is the intelligence that apparently has territorialized the center of our moon and has been working there, as I understand it, for millions of years. And if the government knows all of this, which many people have told me is exactly the case, they know. The second part of your question, which type? You ask about lizards. I would like very much to have hard proof, hard facts, hard photographs, hard videotape, have the whole taxonomy of all of the different intelligences that have been drawn by people in the human abduction syndrome, as well as people who have worked in the military. There were a lot of guys in Vietnam who encountered what they said were shape-shifting reptiles in the jungles of Vietnam. But I've never seen a hard photograph or a hard videotape. So exactly which intelligence would own and operate the center of the moon. I'm not certain. There is a book. I recommend all of you get it. It's 850 some pages. But the longer time goes on, the more I think back about that book, and it's the Brian Scott case. And Brian Scott was an incorrigible teenager going all the way back to the middle of the 1970s when he was out shooting guns. And the short story is his life gets turned upside down as an incorrigible teenager who liked to shoot guns by different non-humans. They interacted with his life that day. And some of the people that he interacted with were the tall, cotton sheet white skinned with red hair, copper hair. There were grays. There were elephantine, like the texture of an elephant, and large bodied. And if you took all the 850 some pages, which they really are worth reading, his whole, I guess, because I talked with him a lot. He died, but back in the 80s and the early 90s, I got to talk with him a lot about Peru and his experiences there, and all of the things that he was troubled by and wanted the world to know, and are now today, sadly, after his passing, becoming more and more acceptable. But he said, had drawings, tremendous detailed drawings, illustrations, that the moon, that's who I first heard it, the moon is hollow, it is filled with what we would call computer equipment. He said it belonged to the gray types, which is a menagerie. There are so many different types. And now we know in 2020 that so much of what's called the gray being world there, it's artificial intelligence. They come in vast array of types. But that was who he thought had hold of the territory at the center of the moon. And I remember that after the first discussion with Brian Scott, that I was left with this kind of refrain, the hollow moon rising like an eye of stone, always watching, always listening to the earth below. I think there's a lot of truth in that. Let's see. I think we're at 8:25. I got Brad. All right, 8:25. And what I want to do is I want to go back now to Hannah, full screen, no edit, no cover, so that we can see what she was reacting to. So Brad, roll that video.
That's not gross shit, though. What
What?
I'm about to do is gonna be gross. That wasn't gross, bro. What I'm about to do is gonna be pretty.
Let's see what you do.
I can't watch. I can't watch it.
Okay, you'll have to.
Told you.
You're so good.
Wait!
Don't fall over.
I told you it was gonna be gross.
I can't watch. Let's see.
You want to come with mommy?
I'm just kidding.
Her dad, John, is a great, great cook. And he would have made a great dinner, but that was the first time that Hannah had ever seen what was going to happen with fish that she caught with her dad in a delightful trip. And it's kind of like another one of those steps in life. I love life. I don't want to do anything to hurt anything, and yet humans have lived in that collaborative combination with the fish world for eons. And I am eager to hear back from you guys whether that really made you laugh, because it was so honestly Hannah just not being able to her reaction I thought was perfect. And Peggy, did anyone guess the right answer?
We did. We had a lot of people guess the correct answer. But the first person who answered it correctly was Roz Hona. I hope I'm pronouncing your name right. I'll put something in the chat here in a moment. So Roz Hona, congratulations.
Oh, thank you. And Roz, I will autograph Mysterious Lights and Crop Circles and get it off to you this week. And I'll do this periodically with things. I think this is fun. And as just a note about Mysterious Lights and Crop Circles, I get so many letters from a lot of you asking me questions about geometry and fractals as the mathematical language of our entire universe. That is the underpinning, the foundation throughout Mysterious Lights and Crop Circles. And in fact, the famous astronomer Gerald Hawkins, he and I knew each other very well. We spent days, hours talking with him. And he was a contributor to my book in an investigation and a discovery that he made about some of the mathematical translations to letters, meaning letters like we would think of an alphabet, but mathematical translations that he found in the crop formations. And it so provoked him, after all the work that Professor Hawkins had done on Stonehenge, that he went on to do a lot of independent investigation. And there is this wonderful, wonderful paper with diagrams that he put, or let me put, in Mysterious Lights and Crop Circles. And I don't think that a lot of people understand how rich my fourth book is with the whole issue of mathematical language, crop formations, and ancient knowledge. So congratulations. I'm eager to hear from all of you about tonight that was just like the world we're in now, a pendulum that went from the light to the beautiful, to the serious, to the challenge of COVID, and ending up back with my beautiful, funny 5-year-old granddaughter, Hannah, and her very, very honest reactions to those fish in the sink with her dad. Thank you for being with me tonight, and I will see you and look forward to being with you next week.